Okay, well, we should get started. So our first speaker is Arpita Idya, and she received her PhD from UCLA, where she worked with David Jaspy, and she's currently at Yale University, where she's working with Menachem Elamilek. And the title of her talk is Membranes for Environmental Sustainability and a Circular Economy. Okay, so Arpita, you should be able to share your screen and start whenever you're ready.
Okay. Okay, great. Also, I want to, before I start, I want to recognize that Ileana also did her PhD at UCLA and we overlapped. I don't know if you remember me, Ileana, but nice to see you here. Okay, so hi everyone. My name is Arpita Idya. I'm a postdoctoral associate here at Yale University. My talk today is going to be about membranes for environmental sustainability and a circular economy, a topic that I've been working on since my PhD and now also in my postdoc. So I want to first start by acknowledging the food, water, energy nexus, which basically highlights the complicated interdependency of each of these sectors on each other. In terms of water, climate change is disrupting global water availability and distribution. We're leading to a declining surface water quality and ocean acidification. In terms of energy, we want to slowly transition away from a fossil fuel dependent one to a renewable energy dependent one. And this transition relies on availability of certain critical metals and elements, which is crucial for the circular, which is also really important. And then finally, in terms of food, I would like to highlight that agriculture accounts for about 70% of the global freshwater use, and enhancing or recovering water can alleviate water scarcity in a lot of regions. But also recovering nutrients from the agricultural wastewater can help reduce our energy consumption, especially that goes towards producing those fertilizers that are that can eventually be recovered from this wastewater. So currently, the way we use our resources is extract it, produce our material, consume them, and then generate waste. The principle of circular economy basically states that we should extract resources from our waste itself to complete the cycle and minimize our actual waste production. So this could be in terms of food, water, or energy. And by that, I mean recovering nutrients from food and agricultural wastewater that will go back into food production, recovering water, clean water itself for drinking or basically reuse, and then recovering critical minerals from either industrial wastewater or unconventional brines so that it could be used for the energy economy. So my research has been promoting circular economy, and my work has been at the molecular scale, where I study molecular interactions within membranes, such as the ion-ion interactions or ion-membrane interactions. My work has also looked at the microscopic scale, where I work on mass transfer processes, membrane interfaces, and even work on developing mechanistic mathematical models. And this is something that I'm working towards, which would be working on macroscopic scales, such as process systems modeling and membrane technology enhancement, or even system enhancement. So I want to first start by talking about my work that I did as a grad student at UCLA, which was focusing on the food-water nexus. So here, the food and agricultural wastewater is a resource-rich stream. It is high nitrogen and phosphorus loading, and if it's not remediated and directly let out into water bodies, can lead to eutrophication and a decline in water quality and aquatic life. Recovering these nutrients before dumping them can help more than just mere removal. And membranes are critical to this resource recovery because of their lower environmental footprint and their little to no chemical usage. So when I talk about this, I would like to first highlight one of my flagstone projects that I did as a grad student at UCLA, which was developing an ion selective membrane for phosphate. Currently, or at that point, where there were very few membrane technologies that existed that were highly selective for a particular species. And transport mechanisms across most commercial membranes is governed by one of two mechanisms. One being size exclusion, where the smaller ion is passed through the membrane and the larger particles are received, or by charge transport, where Donan exclusion prevents ions of similar charges as that of the membrane being repelled and smaller opposite charges being transported across. So we wanted to develop a membrane that can differentiate between ions of similar size and charge. And for this, we looked at the principles of facilitated transport, which basically means we are combining solution diffusion mechanism with that of a complexation reaction. And to achieve a facilitated transport membrane, we add extractants to a membrane, and these extractants act as selective entities, which selectively bind with the target molecules and transport them across the membrane interface. And the complexation is similar to adsorption-desorption, where it's adsorbing on one side, moves across the membrane, and desorbs on the other side. So with this principle, we developed a mixed matrix cation exchange membrane, which consisted of, sorry, which consisted of manganese, hydrous manganese oxide nanoparticles, which were added as dopants. These manganese oxides formed outer sphere complexes with phosphate, which basically means they have slight covalent characteristics, but they would allow selectively binding with phosphate and transporting them across under an applied driving force, and would not form similar bonds with any other anion in solution. The backbone, which was of a CEM, allowed for counter ions to transport across. All the other ions were ejected, and phosphate was selectively transported across the membrane. So when we developed our membrane, we used a commercially available CEM, and we impregnated it with hydrous manganese oxide. Our EDX data showed that manganese was found everywhere inside the membrane and evenly distributed. And performing TEM showed us that the manganese nanoparticles were about 79 nanometers in size. We conducted tests in electrodialysis-like setups where we applied an electrical potential gradient of about 2 volts. Our feed solution on one side consisted of sodium chloride, sodium sulfate, sodium nitrate, and sodium phosphate, whereas the permeate solution on the other side, which was supposed to get the transporting ion, was just the air water. So in an unmodified membrane, we see that it's not selective towards any ion, and we see very little increase in chloride, if any. But in the membrane that we developed, we see that phosphate concentration increased steadily throughout, and all the other ions were equally rejected. So the approach we used of making a facilitated transport membrane using outer sphere complexation helped us develop a membrane which was highly selective towards phosphate and gave us separation factors that went up to 100 depending on the counter ion that we were looking at. And the novelty of the study also lay in the fact that it transported an extremely bulky ion such as phosphate selectively over smaller ions in solutions such as chloride and even nitrate. The impact of this study showed us that we could develop a membrane with extremely high selectivity, and the chemistry of outer sphere complexation is tunable and can be actually applied to recover other ionic species in solutions such as lithium, gold, or even uranium. This paper was published in Nature Nanotechnology in 2022, if anyone is interested and would like to read or know more about this project. The second project I would like to highlight, which is also a study that I'm doing at the microscopic scale, would be at the water energy nexus. This is a project that I'm currently working on as a postdoc here. To give you all a bit of a background, by 2035, we expect 50% of worldwide vehicle sales to be electric. And we expect the demand for lithium is supposed to grow beyond what can be attained from conventional sources, such as mining or saltwater brines. And we need sustainable solutions to extract lithium from unconventional brines like geothermal brines or even battery recycling. So we wanted to study in this project, we wanted to demonstrate a unique class of membrane materials for aqueous separations, and we selected solid state electrolytes, specifically lithium transporting naphthalons, to see if they can provide high selectivity over ions of similar size and charge. In our study, we found that the SSE demonstrated high lithium flux compared to competing ions in solution. So we had a feed consisting of sodium and lithium, and the permeate consisted of no lithium. And then we saw that the lithium concentration continuously increased for up to 50 hours, and absolutely no sodium was seen in the receiving side. We did see one of the drawbacks would probably be that the presence of competing ions and increasing ratios of the competing ions did decrease the lithium flux. However, the selectivity and the separation factor, which is the transport of lithium, was always maintained over other competing ions in solution. So the overall impact of the study that I would like to highlight is that this fundamental study demonstrates near infinite separation factors in solid state electrolytes for lithium over competing ions. And this is a paper we're currently working on, and hopefully we will publish it soon. Next, I would like to highlight one of my studies at the mesoscopic scale. This brings me back to my work that I did at UCLA, which was working on ammonia recovery. So currently, there are several methods of ammonia removal, but most of them rely on bacteria using such as nitrification, denitrification, or even anammox, which is highly temperature dependent, loading dependent, and more importantly, both of them lead to recovery of ammonia as nitrogen by converting them into nitrogen gas. And what we wanted to do was use ammonia stripping to recover ammonium, or nitrogen as ammonia itself, so that we don't have to produce nitrogen gas and then use the energy intensive Haber-Bosch process to convert them back into ammonia, which is then going to be used as a fertilizer in the agricultural industry. So for this, we need to understand that nitrogen exists in wastewater as ammonium ions, and these ammonium ions exist in a pH-dependent equilibrium with a pKa of about 9.24. So above this pH, the ammonium ions are more likely to exist as gaseous ammonia dissolved, and below this, they would more likely to exist in their ionic form. However, this requires large amounts of alkali to increase the pH enough to get to this level so that they could exist as ammonium ions. So we wanted to make our life simpler by using electrochemical methods and split water. When we apply a potential and we lead to water electrolysis, at the cathode, we end up generating hydroxide ions. And at the anode, we generate oxygen or probably even protons. But at the cathode, the increasing hydroxide ions lead to a pH change that shifts the equilibrium towards ammonia existing as gaseous ammonia. And this ammonia can then be extracted using a gas permeable membrane. So with this hypothesis, we developed a process where we created our own, we used a store-bought hydrophobic membrane and coated them with carbon nanotubes to make them electrically conductive. And then use them as a cathode in our setup. For the anode, we used a platinum mesh that would act as anode so that it does not oxidize during the reaction. On the back side of the membrane, we tested either an acidic solution or a vacuum to see which process would recover more ammonia. So to give you basic chemistry of what is happening at the interface, I'd like to talk about when we have a system like this, where we have the cathode, which is the gas permeable membrane that we just created, and the platinum anode, and we apply potential, the water will split to form hydroxide ions at the cathode and protons at the anode. Using a cation exchange membrane prevents hydroxide ions from leaving the cathode chamber, thus increasing the pH in this region. However, it helps the transport of ammonia from the feed solution in the anode chamber into the recovery solution in the cathode chamber. As more ammonia is added to the cathode chamber and as the pH increases, this ammonia is then converted to ammonia gas, gaseous ammonia dissolved, which can then be recovered by creating a vapor pressure difference across the gas permeable membrane. So to study the process itself, we wanted to see if the process is either limited by current or if it's a mass transport limited process. So we did that by making 3 different types of membrane, one with just carbon nanotubes coated on them, and 2 more with by coating nickel on the membrane. And why nickel? It's because nickel helps reduce the water electrolysis over potential. So we added nickel, we deposited nickel for 6 hours to see if that changes the membrane properties. And then we grew nickel for a much longer time to see if more nickel would help us generate more current or help reduce the water oxidation, the water electrolysis or potential. So here we studied the mass transfer across a gas permeable membrane, and we know that is affected by either vapor pressure difference across the gas permeable membrane or the membrane properties itself. We wanted to study ammonia recovery, which would be across the gas permeable membrane recovered in the recovery chamber, or the ammonia removal, which would be ammonia removed from the feed solution in the anode chamber and then recovered in the recovery chamber. So when we look at current efficiencies for the three membranes, we see that the current efficiency of ammonia removal, which would be ammonia removed from the feed chamber across the CEM, was pretty low, about 0.1 to 0.3, meaning that the ammonium transport across the CEM is limited by current. So for the amount of ammonia present in the feed solution, we could be working at higher potentials to have a higher current across, to have more ammonia removed from the solution. However, when we look at the amount of mass transport across the gas permeable membrane in all of these cases, we see that it's always much lower than that of the CEM, meaning that ammonia recovery itself could be mass transport limited. We then wanted to study the different applied methods for ammonia extraction, which was acid solution, by either circulating an acid solution on the recovery side or by applying a vacuum to recover ammonia as gaseous ammonia itself. And we see that, so by ammonia removal rate, I mean the ammonia transported across the CEM and removed from the feed. And by ammonia recovery rate, I mean the ammonia which is recovered in the feed through the acid solution or the vacuum across the gas permeable membrane. So here we see that when we apply an acid solution, we have a much higher ammonia removal rate and a much higher ammonia recovery rate. When we use an acid solution, we're able to recover about 63% of ammonia, whereas when we apply a vacuum, only 30% of ammonia is recovered. And this is because ammonia has a really high Henry constant, which leads to an extremely low partial pressure. So when we apply a vacuum, the amount of vacuum we would need to apply would be beyond what the membrane can take. And in fact, it was beyond what we could achieve with the vacuum pump we had in the lab. However, when we use an acid solution, because it had no ammonia present, the vapor pressure of ammonia in the acid solution was basically next to zero. So we were able to recover a lot more ammonia when we use an acid solution as the recovery method. So this shows us that ammonia recovery itself is mass transfer limited, and acid recirculation had a much higher driving force for recovery. So overall, the impact I would like to highlight with this project was that we successfully extracted and recovered nitrogen as pure ammonium sulfate from wastewater by using an acid circulation stream, which was sulfuric acid. And we improved the understanding of the process, which would help us when we want to look at process intensification or building up a larger plant. So that brings me to my last slide of acknowledgements. I would like to thank David Jaspi, my professor at UCLA, and my lab mates that I worked with. And I would also like to thank Professor Menachem Elimelech, my advisor here at Yale, and all my lovely lab mates that I worked with. And last but not the least, my family. And that's it. Thank you.
Thank you very much. That was very interesting. We have about 12 minutes for questions. So yeah, so Ileana, do you want to go first, Ileana?
Oh,
do you have your hand raised or are you clapping?
It's clapping.
Okay. So why don't you go, Lee? And then I think there's enough people, not that many people. So if you have a question after that, you could just ask your question.
All right, sounds great. So first, thanks for an excellent seminar. I really enjoyed hearing about your work.
Thank you.
I wanted to ask a couple of questions about the first part with the phosphate recovery.
Yeah.
I was really interested in it. It kind of reminded me of some of the research I started doing as an undergrad that got me excited about environmental engineering in the first
Before
place.
you ask, I just want to mention that in 2022 at ACS in San Diego, I had given this talk and you were the chair of that session. And I had given this exact talk to you then as well.
That's okay, that's okay. We'll
Um,
see how consistent I am. Maybe I'll ask the same question. Um, But, back when I was getting started with research, we were looking at developing kind of hybrid ion exchange resins, for treating arsenate and phosphate, and things like that. And so, I really liked the first part of your talk, which was kind of just bringing it into the membrane,
Mhm.
form, maybe for similar purposes. But I'm curious, I know you used cation exchange membranes.
Yeah.
Did you explore anion exchange membranes as well, or no?
So we did not, because the reason we went with cation exchange membranes is because it would be inherently blocking all anions, and then we wanted to impregnate them with specific extractants that were phosphate specific to see if that was the hypothesis we wanted to go with, to see if that would allow for phosphate recovery, and it did. Had we used anion exchange membranes, we would have had to look for extractants that would block all other anions and not form any bond with phosphate to allow phosphate through. But that's a great avenue of research that we could actually look at next.
Yeah, I know one thing. So I should mention, so my undergrad advisor is Arup Singh Gupta, and
Yes.
so really all of these ideas are his, not mine. But I know when they were kind of looking at, arsenate in the first place, they saw better performance with the hybrid anion exchange resins compared to the hybrid cation exchange resins.
Mhm.
And I'd have to kind of look back, I think the, intramembrane transport is maybe a more important, aspect of your work compared to the
Mhm.
resins, but pretty interesting. And then second question, I know the hydrous manganese oxide, also has some redox properties.
Yes.
You take advantage of those in some way with your hybrid membranes, or not really? It was really just kind of as adsorbent steps.
It was just as an absorbent. In fact, not yeah. So we did not take advantage of its redox properties at all, actually.
Cool.
Yeah.
Well, thanks so much.
Thank you so much.
I have a question, or
Yeah.
so I was surprised when you said we wanted to make our life simpler, so we did water electrolysis. And
Mhm.
so as a practical matter, as it compared to just adding a caustic like
Mhm.
sodium hydroxide, as a practical matter, do you think there's any advantages to that electrochemical production of hydroxide compared to using a chemical reagent to raise the pH?
I would say waste generation, because eventually we have two streams when we are splitting water. One that generates a caustic stream, the other one that generates an acidic stream. And eventually, because we have two streams, once we are done with ammonia recovery, we could maybe neutralize the stream back, and then not have to deal with the chemical waste, as opposed to using an alkali itself, where we'd end up with the stream that has a really high pH, and then we have to use more chemicals to neutralize it before we get rid of the water. That would be one of the first things that come to mind.
Okay.
But
Okay.
yeah,
I guess it would be interesting to really look at the costs, as a
yeah,
practical matter. I do have another question if nobody
yeah.
has a pressing question, and so I'll just ask, which is, what was the mechanism for lithium selectivity in the ion selective membrane in the second part of the research
Yes.
that you talked about?
So we used a ready-made Nasicon, which is a solid-state electrolyte that is used in batteries. So the way they work is intercalation. So it's very size and charge specific. The structure is, I'd have to go back. I've had to ask you to look at the chemistry you probably studied in your first 4 years of engineering, which would be the structure of the unit cell inside the Nasicon itself is designed in such a way that it's electrically neutral and it already has lithium. So by intercalation, it would be, and when a lithium enters, you have a lithium leaving on the other side, and then all the other lithiums within the structure itself migrate to the next position. So the structure itself is highly lithium selective, and it does not allow sodium in because sodium has a slightly higher ionic radii, and it does not allow magnesium in, because even though it has a similar ionic radii, because it has a slightly higher charge. So it's a very well balanced structure itself. So it's a very materials research intensive um, um, electrolyte, I would say. So I don't know if that
So is
answers
it
your
like a
question.
solid material? You said
Yes,
you,
it's a ceramic membrane is how I want you to imagine it. And the structure itself is very rigid, and it's basically like a unit cell, and then you multiply that across to make a ceramic membrane. And we used the ones that were commercially available, and we were just studying them to see if they would work. And most solid state electrolytes, unfortunately, are not stable in water because they have to work in batteries. So they have to be in vacuum. And they're like, you make a cell with the lithium electrode on one side and the intercalation cathode on the other side with the solid electrolyte. And they're all sandwiched together in vacuum. So then there's no air or no water. So we wanted to study to see if we have a solid electrolyte that is good enough to work in a water basis that is water stable, first of all. And if it is water stable, what are its performance metrics? Do we see it as a promising avenue of research or applicable research when it comes to recovery of lithium in wastewater streams?
okay, thank you.
Thank you.
So what other questions do we have?
Yeah.
I'll come back again.
Yes.
So back on the first part.
Mhm.
Yeah, I know you mentioned that you use the cation exchange membrane to kind of block, the other
Yeah.
anions. Can you just remind me what kind of agricultural wastewater were you focused on there?
So we did not test it with actual streams. We were looking the testing feed solution that we had equal concentrations of sodium phosphate, sodium chloride, sodium sulfate, and sodium nitrate. We never ended up testing them on actual agricultural wastewater streams.
So I'm just kind of thinking out loud here. So like
Yeah.
if you had, let's say, some calcium, in like a animal manure
Hmm.
wastewater or something like that, would that calcium transport across the membrane?
Yes. So because we're using a membrane with a backbone that allows for cation transport, all cations would go through. I believe because of the high sodium concentration, we can have selectivity between sodium and calcium depending on the current densities we're looking at. But our study was a very fundamental one just to see if the hypothesis works. Um, and yeah, and then so to answer your question, yes, calcium would definitely go across. Most cations would.
So maybe just, one idea, and I don't know how to do it, so I'll just throw this out for you. We've done a lot of work kind of thinking about phosphate recovery from animal manures, and a
Mhm,
lot of animal manure, with the exception of poultry, has too much calcium in it. That kind
mhm,
of the thermodynamics, the recovery system kind of lends towards hydroxyapatite
mhm,
precipitation recovery, which is not preferred. Like usually we're trying to produce struvites or
mhm,
something else that has a little bit more value. And so if you could find a way to tune the selectivity, for the cations as well, to
mhm.
prevent Ca2 plus transport in
Wow.
particular, I think that would be really cool and, help to address, maybe some of the downstream concerns if you're recovering both calcium and phosphate.
That is actually a great research topic. Thank you so much.
Yeah, sure. I know that was like the minor part of your presentation, but
No, but this
I
is
was, I was
a
very
really
intrigued, so I
interesting
enjoyed it.
avenue to have. Thank you.
What other questions does everybody have? Okay, I'll ask another question, which is but the question is, what kind of flow rates were you able to achieve? Or is it practical to achieve with these kind of membrane
Uh,
technologies?
which specific project were you looking at?
The best one, the one you could achieve the highest, flow rates of.
So I think all of these studies, we used a batch system. So we were not circulating we were not circulating the solution. Having said that, when it comes to ammonia recovery and using an acid circulation stream to recover ammonia, I believe that would be our best bet to get continuous ammonia recovery and at high enough rates that we could actually scale that process up to and maintain its energy efficiency at, say, a pilot scale as well.
Okay. Okay. Well, thank you.
Thank you so much.
So Jessica Wilson is here from Manhattan College, and she's going to introduce our next speaker. And I would like to thank Arpita for
Thank you
a
so
really
much.
interesting talk and a great Q&A. So thank you very much.
Great. Thanks, Liz. So Ileana, you can start sharing your screen while I introduce you. So Ileana Callejas is a postdoctoral fellow at NASA Jet Propulsion Lab and is also a climate resilience fellow at UCLA Center for Diverse Leadership in Science. And she also, her third job is an assistant professor at Biola University in the biological sciences, and she completed her PhD at UCLA. And so her talk is Emerging Techniques in Coastal Water Quality, Metagenomics, and Remote Sensing. So thank you, Ileana.
All right, yes. Are you all able to just see my slides right now? Okay, great. All right, well, thanks everyone for coming today. I just want to share my research and from my doctoral degree and then what I'm working on now as a postdoc at NASA. And basically this idea of looking at coastal water quality through a bunch of different tools from metagenomics to remote sensing. And so just to give a little background myself, I'm a triple Bruin and my background is in environmental science and environmental engineering. And so as through my introduction, you can see that I've been teaching a lot in biology departments. And right now I'm at the NASA Jet Propulsion Lab in the Water and Ecosystems group. And so it's a really great place to see how people are leveraging all of these different satellite remote sensing tools to look at water resources and water quality. And so here's just an overview of my research. My research has been very interesting, especially since I did my PhD during COVID, which ended up leading me to have a bit of a different path and I guess, again, in different research skills. And so one of them is in environmental microbiology, public health. And then during COVID, moving more to satellite remote sensing. And I'm also really passionate about environmental education and how that can be used to inform my research. And so first, I want to talk about what I started off in my master's thesis and in my PhD at UCLA, which is more on environmental microbiology and public health, really focus on the fate and transport of emerging contaminants in the environment. And so here, I just want to give the statistic that over 2.8 million antibiotic-resistant infections occur in the United States. And as a result, thousands of people die. And so antibiotic resistance is a really pressing global health issue. So it's, if anyone has heard of the word superbugs, and so it's this idea that bacteria are resistant to antibiotics. And so there always needs to be more antibiotics or different ways to treat these infections. And it places a lot of burden on the health system. And so there's a lot of different ways that antibiotic resistance can be moved throughout the environment. And so some of it can be through livestock, especially since we give antibiotics at low doses to livestock for growth promotion and for them not to get sick, but that can end up in the manure and in the soil. And that was actually part of my master's thesis, was more looking at the antibiotic resistant gene levels in commercially available soils that you can get from Lowe's and Home Depot. For my PhD, I moved more to this side where we can see that there's a big anthropogenic sourcing of antibiotic resistance as well through the healthcare system, and then passing through the wastewater treatment plants, which can then end up in surface waters, which is where I focused a lot of my work in this area, mainly looking at rivers and in beaches for antibiotic resistance. And here is just a review of all of the research that is currently being done in coastal antibiotic resistance. And so you can see there's a lot more being done in places like China and in Europe. some places here in the US, virtually nothing here in Central America. And so that's also a big issue as well. And so one of my first projects that I started off as a new PhD student was just looking in my area in Los Angeles. And so with the help of a lot of undergrad students, we were eventually able to take samples all across the Los Angeles River watershed, in some places up here in the mountainous areas, all throughout the developed areas of Los Angeles, and also here in some beaches. And the interesting thing about the Los Angeles River is that a lot of the flow is actually from water reclamation plants. And so we also wanted to see above and below those treatment plants to see what was the antibiotic resistance loading from those places. And so what we saw overall was that in terms of the fecal indicator bacteria, that the wastewater treatment plants were actually diluting the fecal indicator bacteria, which is not surprising as the water from the reclamation plants are treated to a very high level, but not necessarily through the antibiotic resistance gene. And so here, one of our highest areas that had antibiotic resistance genes, as revealed through the metagenomic sequencing, was an area that was below one of the wastewater treatment plants. And so this is currently under review and should hopefully be out soon. And now, if you remember that map that I showed, the antibiotic resistance is also almost like an environmental justice issue as well, because it disproportionately affects low and middle income countries, especially due to maybe like less access to hygiene, clean water, and maybe less regulation on the antibiotics itself. And so low and middle income countries are disproportionately affected. And also in terms of the research, as we saw from that map, there's not too much research being done in the global south in these areas. And in this case, we also were influenced by the World Health Organization's global tricycle surveillance. And so that's a protocol specifically for E. coli that's resistant to beta lactamases. And so their recommendation is to look at some of these key hotspots for antibiotic resistance. And so one of them being places where there's a lot of human waste, some wet markets, and then upstream and downstream of major rivers. And so after COVID and on all of the international restrictions, a team of myself and other scientists and other graduate students were able to go to Belize and actually focus in on taking some samples in the Belize River, which is one of the major rivers in Belize, and specifically look at Belize City, which is one of the most populated cities in Belize, in some of these key areas. For example, poultry operations and an open fish market. And then here outlined in the yellow is a water treatment lagoon system where we also took a sample offshore. And so what we were able to see here that there was high levels of antibiotic resistance within this Hollover Creek that's going through the city, particularly in this fish market, which you can also see here as revealed through metagenomic sequencing. And we also were very intrigued to also look at some of the coral reef areas. There also hasn't been too much research done in antibiotic resistance in coral reefs, so we were also able to see that. And though the levels were lower, they were not completely devoid of antibiotic resistance genes. And this paper has also just recently been submitted. And just to go a little bit deeper, looking more at the Belize City with some of this culture-based methods, we were able to see that indeed that this fish market had the higher levels of this cultivable antibiotic-resistant E. coli. But surprisingly, this area over here close to the wastewater treatment lagoon system didn't actually have a presence of these viable antibiotic resistant E. coli. And so that was very interesting to us. And our partners in Belize were also very excited to hear about this as well. And so now during COVID, could not do a lot of this wet lab research. And so I actually ended up pivoting more to using satellite remote sensing. And so these are all of the satellites that are in space. And so here I just have a little bit of an overview of just NASA's Earth observing fleet. And so some of these that I have used are, for example, this Aqua. And these are some of the instruments that are on the International Space Station. So some that I have used before and are using currently is EcoStress, Emit, and another really great one that just launched this year, which is PACE, which is specifically focused on phytoplankton. And so with these tools, the idea is this, that you can look at an image of water, and let's say you have certain pixels where maybe you can detect more chlorophyll presence or more color dissolved organic matter, maybe looking like more of this black color, or a higher reflectance, looking at the non-algal particles and sediments. And so by looking at different pixels, you can see that, for example, with the sediment one, you would see a much higher reflectance. It's going to have a much higher remote sensing reflectance signature among the different water pixels. And if you're interested in learning more about this, I actually have a very brief little paper in Nature Reviews Earth and Environment that's all about learning how to monitor coastal water quality with the satellite data. And I think another really cool aspect about this is actually bilingual article, and that's with the help of someone from GeoLatinas. And so during COVID, I got started as an intern at NASA Jet Propulsion Lab, and I got started on this project, which is the Belize Sustainable Development Goal Project, which was actually one of the first Belize-specific NASA research projects. And so the idea here was to utilize not only NASA data, but also the European Space Agency data to monitor the marine environments and how could we develop these national monitoring methods, and also engaged in some technical exchanges. And so you can see one of them was down here. Since this was COVID time, a lot of it had to be done online until we were eventually able to meet once the closures were done. And the main motivation of this was to actually develop policy recommendations based on the United Nations Sustainable Development Goals, specifically number 14, which is life below water, and 15, which is life on land. So I've been talking a lot about Belize, but maybe I haven't given too much of a background, but they have a lot of really beautiful coral reefs, which has a lot of benefits through coastal protection, fisheries, and also tourism. And a lot of the public depends on the marine resources and the coral reefs in some way or another. And so one of the first projects that I worked on was during COVID. And so we were very interested to see, since this is such a touristic place, a lot of marine traffic, how did the decline in the marine traffic during COVID affect the water quality as a whole throughout the coast of Belize? And so I used commercially available marine traffic data, and I used satellite-derived KD490, which is basically just a proxy for turbidity and water clarity. And I also used some runoff and precipitation models. And so, for example, here are some of the maps that I was able to produce that's comparing 2020 to a 2002 and 2019 baseline. And so if we remember, for the most part for COVID, a lot of the international shutdowns occurred at the end of March. And so going through some of these maps indicated by the blue color, we can see that there was a recovery in the water clarity throughout most of the months in Belize, except for November, where there was actually two really large Category 4 hurricanes that hit Central America. And so that brought in a lot of runoff and precipitation. And so we were able to use the satellite remote sensing to detect these improvements and also this deterioration during these extreme events. And this was actually input into a case study into the Belize Interim Coastal Management Plan. And this paper is already out if anyone is interested. So following on this, we didn't just want to look just at the turbidity, but we also wanted to incorporate sea surface temperature as one of the key metrics for looking at coral bleaching. And so the idea is that if there's this really long sustained periods of time of sea surface temperature, that the corals can bleach. And so we looked specifically at the marine protected areas across the coast of Belize, and I created a coral health index to rank the marine protected areas by both of these two metrics. And so I was able to rank them, and we were able to give this to our partners, and they weren't surprised to see that one of the port areas down here in the southern area was the one that was most stressed in both the turbidity and the sea surface temperature. And we were able to see that that was the case for a lot of these marine protected areas that are a lot closer to the coast. And this workflow is also available on my GitHub, and it's also published online. A lot of it was through use of Google Earth Engine and RStudio. And so what I'm working right now throughout my postdoc, we actually just got funded through an internal grant between Caltech and NASA Jet Propulsion Lab to continue this work and expand it even further. So on the JPL side, not just looking at Belize, but looking at the Mesoamerican region in general, and also expanding to the Coral Triangle, which encompasses many of the countries in Southeast Asia. And then we're also working with a professor at Caltech, who is a professor in environmental economics, which is going to take the maps that we are producing and looking at household survey data that has a lot of really interesting indicators, such as electricity, access to toilets, and even utensils, to see what is the long-term effects of the coral bleaching on societal well-being, and to just try to merge these together to create solutions across these different disciplines. And so what I'm working on right now in my postdoc is using this global coral bleaching data set, which goes back from 1985 to 2020. So there's over 40,000 reef surveys, and I'm comparing that right now with NOAA Coral Reef Watch bleaching alerts, which is on a 5-kilometer scale. But there's some areas where maybe those pixels are a bit too large to resolve some of the thermal differences in the water. And so I'm using NASA EcoStress data, which is thermal data mostly used for land surface temperature and evapotranspiration, but looking at its ability to look at sea surface temperature. And so this is on a 70-meter scale. And so you can see this is an EcoStress scene, and it's able to capture some of these differences on these really small atolls, but that's where the coral reefs actually are. And so finally, moving on to my third area of research, which is environmental education. And so while I was TAing at UCLA, I was working with my advisor to implement remote sensing modules into some courses at UCLA. And so one of them was in an upper division environmental engineering course, which is chemical fate and transport and aquatic environments, and in another course that was non-general first-year students, mainly students that were for the most part non-STEM. And so we were able to do some modules with them in class, looking at just using Google Earth Engine to look at some different environmental issues such as sea ice and also deforestation. And we were able to survey them before and after these modules. And so we could see that a lot of these gains were what we saw among the students was mainly in the skills. And so that's coding, editing coding, and also an understanding of remote sensing. But we didn't really see too many gains from these modules in terms of interest in science and an interest in a career in environmental research. And that was both across these non-STEM students and the STEM students. So it was probably too short of an intervention. But we did take it a step further with the STEM students in particular to do course-based undergraduate research experiences. And so they took that first question that I had, that was, what happened during the COVID pandemic? What were the gains in water clarity during that time? And so we formed them into randomized student groups. And we were actually really impressed to see that a lot of the students were actually able to look at these different areas and were actually able to see some differences. And so one student group looked at the sport area in Bangkok, Thailand, and were actually able to see that there were improvements in turbidity during 2020 as compared to this 2018 baseline that they had. And so all of these results are already published, and it's just really interesting to see. And I could really see this being as a tool to also inform the research that we do in our labs and in our groups. And so just for the future, and what I'm also doing now is to try to bridge and to merge how can we use the microscopic and also the data that we're getting from space to really understand our environment and the fate and transport of pollutants. And so one of the other projects that I'm working on right now is using in situ data and remote sensing data to look at the Tijuana River, which here in Southern California is notorious for basically consistently closing down the beaches because of all of the sewage that is being passed through this river. And so I'm looking at some in situ sequencing data from some collaborators at UCLA and also some fecal indicator bacteria. And I'm using NASA EMIT, which EMIT is an instrument on the International Space Station, originally used for mineral sourcing for dust, and PACE data, which is newly launched, mainly looking at phytoplankton, and was able to produce some of these maps to see some of these plumes, and also trying to see what are the matchups between these kinds of scenes and then these in situ data to see how we can look comprehensively at the fate and transport of the sewage and the pathogens through the environment. And so some of the things that we can see, for example, is a high Senecacoccus or phytoplankton that is resulting in some of these coastal areas. And so one of the ideas that I think I have in the future is to not only look at pathogens, but to also look at this other really interesting and up and coming biodiversity indicator, which is environmental DNA. And so this is the idea that animals and other organisms will shed their DNA into the environment, and you're able to monitor biodiversity in that way, in this less invasive manner, and to connect that with satellite remote sensing to monitor the biodiversity and how the environmental parameters are influencing them. And so that's been an overview of my research. And I think another key thing and one of the motivators for me is also this idea of One Health, where maybe as engineers, maybe we're focused more on the human aspect of it, but we're also focused on, we know that we're all connected in some way or other to the environment and also animal health. And so by monitoring all of these things and through all of the different techniques that we have, we're living in a time where there's so much data being collected all the time to really harness that for the well-being of animals, environment, and human health. And with that, I thank you all for paying attention and coming to this talk, and I open it up to any questions and discussion, and I leave my email here in case anyone wants to chat offline.
Yeah, thank you so much, Ileana. So we have some time for questions. Would anyone like to start us off? All right, I'll go ahead, Lee.
On the stage here today. Thanks so much for the awesome presentation. I love how you kind of wove all of these different skill sets, together, from the satellite data to the kind of on the grounds data. It's really impressive to me. Some of the work that our group has done in the past has focused on UV filters, this kind of active ingredients in sunscreen chemicals, and there's some concern that they're toxic to corals. I would say there's maybe conflicting evidence. But do you think it would be possible maybe to use like some of the same wastewater-based tools, to model potentially UV filter inputs, in these areas, maybe especially like the tourist beaches, to see if that also can be picked up with like some of your coral health indicators?
Yeah, I think that's definitely something that I've thought about as well, especially with the rise of stony tissue coral disease. And so I think the only way possible might be if there's also a reflectance in the corals, but there is some caveats as well to the satellite remote sensing, such as if there's no, say, like bleaching indicators where there's a difference in the reflectance in the corals, and that may be difficult. And I think another one that we're dealing with right now too is if the coral is too deep, then the remote sensing imagery is not able to capture it. But there are some people that are taking advantage of unarmed vehicles and drones to also have this more of a deeper, more comprehensive view as well. And so I think that that could be one potential solution.
Yeah, thank you.
I think Liz, your hand was up next.
Thank you. I just have a very basic question about how you interpreted the satellite data to be, for example, phytoplankton. And you said for at one point, KD 490 was a proxy for turbidity. Is that the wavelength, 490 nanometers, like a color?
Yes,
Is
yeah.
it
So
like,
typically, Mhm. Go ahead. Yeah, so the KD 490, that's an indicator of basically how far the light is penetrating into the water. So it doesn't necessarily tell you what the constituents are. So it could definitely be like sediment based, or it could be more algae based. So it doesn't necessarily tell you that. But with some of the more recent technologies that are coming out, especially in the hyperspectral data sets, we're able to see a lot of different wavelengths. And so I think the interesting thing with that PACE satellite that recently came out is that they're actually able to use algorithms to detect different phytoplankton species. And that's just due to being able to resolve some of those different wavelengths within the visible spectrum.
got
And
it.
so KD 490 is definitely more general, but with the more recent missions, such as Emit and PACE, we're able to get a lot more at the finer scale with that data.
Thank you.
Bill, go ahead.
Very interesting talk. Having studied oceanography for my PhD, I love it. One thing I would mention, just this morning, I downloaded a paper from PNAS on carbon-centric dynamics of Earth's marine phytoplankton, and I can send you that reference if you want to just send me an email at wcooper@uci.edu. But the other thing is this antimicrobial resistance. It seems to me that we're going to have to start thinking about that in much more. I mean, it's scary. And we have to start thinking about disinfection maybe in different ways and how we get rid of some of those things. Of course, some of my background is electron beam irradiation, which would be, it's certainly not, it might be expensive, but it's certainly not specific. And then I have another real quick personal question about Belize. That little red blob you had on the bottom, was that Placentia? Yeah, all the way down south in Belize.
It is definitely more south, and so
I think it looks like it might have
it's
been
possible,
Placentia.
yeah, that I think it was closer to Placencia. Yeah, that whole port area. I think that one was a port of
It
Port of
doesn't matter.
Honduras, I think.
Could be. Yeah.
What that was the highest.
When I was there, Placencia was just the main street on Placencia was a 3-foot wide concrete sidewalk. That was the main drag at Placencia. But I think that this antimicrobial resistance is really a scary thing, because we use the microbial resistance is just growing, and I think you had some pretty scary statistics on that. Do you have any thoughts about that? How are we going to deal with that on a worldwide basis?
Yeah, I think it's definitely a big issue, and I think one of the areas is going to have to be its prevention, and so the misuse of antibiotics in the healthcare system and also in animal agriculture. And then also, I think there just needs to be more advances in the research side as well. I think I mean, I guess technically we would probably want zero antibiotic resistance in the environment, but to even have an idea of what is the level that we should attain within our wastewater treatment plants. And I think, yeah, it's definitely very difficult with antibiotic resistance because it's such a dynamic pollutant that if it gets in the right conditions, we've even seen that in wastewater treatment plants, maybe in some areas it's eradicated, but in other ones, the conditions are just right to increase it and accumulate it. And so it's definitely a very tricky dynamic contaminant.
Thank you.
I'll ask a follow-up question to that. So on one of your slides, you mentioned that coral reefs have lower levels of antibiotic resistance. Was that right? So that was lower levels compared to like other areas, or?
Yeah, I guess in that study, I was comparing more to what we saw in the Belize city. And so areas that have, a lot more outputs from, for example, that treatment lagoon, the fish market, being a lot more closer to these land-based sources. And so it could very well be that, it's a lot of these reefs were far from the coastline. And so in some ways, the coastal lagoon kind of dilutes some of that signal.
Mhm.
I think out on the reefs too, you're going to have clearer water, so your photochemistry is going to kick in there as well.
Yeah. Yes.
Getting back to Lee's question about the sunscreens. I mean, I'm trying to think what we use for doing triplet. Lee, what's the chemical that benzophenone is very highly reactive for developing a triplet state. That's what we used to look at triplet state and some other rate constants when we were doing some work. But a lot of the sunscreens have got benzophenone as one of the major components. So I would expect that benzophenone would be extremely photochemically reactive.
So we've come up just at the end of our time here. So I just want to, I really want to thank our speakers, Ileana and Arpita. This was a great seminar, and I'm really happy with the turnout and all of the great questions. And this has been recorded and it will be uploaded somewhere. Liz, is that right?
Let me check. I did record it, and I'll ask the organizers where we might upload it.
That's great. Okay. And so, yeah, so let's just thank again our speakers. And I think we have one more of these seminars coming up, or two more of these seminars actually coming up on the 22nd and the 6th. So be on the lookout for announcements from the Student and Postdoc Services Committee regarding those. They'll be coming out in the next couple of weeks, so we hope you're able to join us for those next two seminars as well. All right, thank you so much, and enjoy your afternoon.
Thank you.